Vi chiedo preventivamente scusa per la mia pronuncia, but it is my great honor and pleasure to introduce the next speaker, T.Chatila, the professor of pediatrics at Harvard Medical School and senior physician in the Division of Immunology at Boston Children's Hospital. Research of Professor Ch.Atila focuses on elucidating the mechanisms involved in the breakdown of immune tolerance, both in monogenic disorder of immunity and in common allergic inflammatory disorders, including asthma and food allergy, with a particular focus on the role of regulatory T cells in disease pathogenesis. His work has led to the identification of several genetic networks that regulate immune tolerance in human subjects, including those involving FOXP3, DOCK8, LRBA, and EL4 receptor genes. Please join me in welcoming Professor T.Chatila, that will talk to us about primary atopic disorders and inborn error of immunity. Thank you.
Thank you. Thank you for the kind invitation. I would be having to use my computer, so I'll be orienting myself a little bit away from the desk. Can you hear me? Yeah. Okay. See if I can get the computer all the way here. Or a microphone. Yeah, microphone's better. Yeah, I can. Maybe I can. Perfect. Thank you. Okay. So the topic I'm talking about today is primary atopic disorders and inborn errors of immunity. And the background for the interest in this subject, of course, is the overwhelming increase in the prevalence of allergic disorders over the last century, even before, dating all the way back to the Industrial Revolution. And what this slide shows, this is from a review by Thomas Platzmill in Journal Allergy Clinical Immunology some time back, is that there has been really like waves of allergic diseases coming in epidemic forms, one after the other, starting with rhinitis, allergic rhinitis, then going to asthma. And the last one that we are still sort of going through is the epidemic of food allergy. And a lot of the work has been done on the environmental factors that promote allergic disorders. Now, this slide summarizes them very briefly. To make a long story short, lifestyle changes associated with the modern lifestyle, with the things that have happened after the Industrial Revolution, including urbanization, sort of hygiene, enhanced hygiene, small family size, antibiotic usage, and so on, all have contributed to increase in allergy. And in fact, like families that live in a very sort of traditional lifestyle in farming have much less prevalence of allergic diseases. The subject of our talk, though, is what are these environmental factors acting upon to promote disease expression in the human population? And I would point out to you that there's a whole block of genetic predisposing factors that make these diseases possible, both in terms of allergy induction, but also specific disease phenotypes. And the subject of our talk today is to dive a little bit more into these genetic predisposition, taking advantage of inborn errors of immunity that people have described and which have been associated with primary atopic disorders. And so a lot of work has been done on primary atopic disorders, especially in recent years. It has lagged behind other work in inborn error of immunity related, for example, to combined immune deficiency or severe combined immune deficiency, if only because people at the time thought less about allergy as a problem of inborn error of immunity and more of severe immune disorders as being so. With time, with the identification of more diseases, sort of different categories have emerged. So we know that there are defects, for example, in the STAT pathway, which I will talk about, which are associated with primary atopic disorders. Others, such as the actinopathies, are also associated, for some of them, with atopic disorders. Problems at the barrier, skin barrier, filaggrin deficiency, SPINK5, and others have been associated, again, with allergic disorders. So the list is too large to dive into it into one talk, and what I will do is, I will go back, I'll lean back on our own work on some of these pathways to give you a couple of examples of how we have investigated these disorders and what sort of lessons we derived from them. So the first sort of part of my talk will focus on defects in the IL-4 receptor STAT6 pathway. And this is a pathway that has attracted my own attention for a long time, not the least because one of the first manifestations of a severe atopic disorder in an experimental mouse model was the creation of what's called the IL-4 transgenic mouse by Phil Leader's group back in 1990. And that mouse had a lot of phenotypes that we associate with the hyper-IgE syndrome, very high IgE levels, skin inflammation, and so on. And so we and others actually have looked into defects in this pathway for quite a while. The pathway itself is composed of dimeric, heterodimeric IL-4 receptor complexes that have an IL-4 receptor alpha chain common to them, and what's called an amplifications chain, the gamma C chain in the type 1 IL-4 receptor, and the IL-13 receptor alpha-1 chain for the type 2. And they both signal downstream via STAT6 to mediate gene expression and activation. And if you just zoom in on the IL-4 receptor type 1, we and others have identified sort of polymorphisms common in the population that are associated with enhanced receptor signaling and association with diseases such as asthma. More recently, there have been identification of inborn error of immunity, rare genetic disorders that affect components of this pathway, including the transcription factor STAT6 and JAK1 kinase. And I will dive in a little bit more into the STAT6 because we and others have published that over the last couple of years. By now, actually several groups have identified gain-of-function mutation in the transcription factor STAT6. And these mutations are not different from what you see in other transcription factors of the STAT family in that they cluster in different domains. In this particular case in STAT6, they cluster in the DNA binding domain in particular with some other mutation in different domains. And it's similar to the IL-4 transgenic mouse in a way. I mean, you end up with disease phenotype that spreads across several systems, including the skin, the lung, gastrointestinal tract. You have very high IgE levels, and you have severe allergic responses in these patients. You also have some non-allergic manifestations, such as aneurysms that you see, and malignancy. In fact, STAT6 mutations have originally been identified in tumors as somatic mutations associated with tumor progression. And some of these patients actually do develop lymphomas. So this, in a way, brought together many of these threads about how this system could be working in terms of promoting allergic diseases and also potentially targeting it therapeutically to treat these patients and prevent progression to more serious manifestations. And this is just, I put one slide to show you how these mutations work. What you're seeing here is an immune fluorescence analysis of cell line, HEC293, that has been transfected with either a wild-type STAT6 or STAT6 bearing a gain-of-function mutation. And what you see is that when you stimulate these cells with IL-4, which is shown on the right side, the phosphostat6 shown in red is much more apparent in the nuclei of the cells, as the STAT6 translocates very vigorously to the nucleus compared to wild-type STAT6, the mutant STAT6 that is. And this enhanced STAT6 translocation is associated with enhanced activation of its gene transcription program in the nucleus. So some of these mutations just make STAT6 translocate, be phosphorylated and translocate better. Some of them make STAT6 sort of transcriptionally active better. So they have different effects, actually, even though they're all gain-of-function mutations. And we thought about treating patients with STAT6 gain-of-function mutations with precision therapy targeting the JAK kinases that turn on STAT6 itself. So in this particular case, we use ruxolitinib, which is a JAK1/2 inhibitor, with the idea of inhibiting signaling via the IL-4 receptor that ultimately feeds into the mutant STAT6 and triggers it to be activated. And in a case demonstration, we treated one patient with STAT6 gain-of-function mutation with ruxolitinib, and he dramatically responded very quickly. Actually, the most remarkable response was the itching. These patients get very severe skin itching, and that sort of disappears very quickly within a couple of weeks after starting therapy. Now, if you look at other stigmata of the disease, like the IgE goes down, but it doesn't go down all the way. Eosinophilia does respond very well to the therapy. Other markers of type 2 immunity do improve in these patients, suggesting that, in fact, you can get sort of persistent response to therapy as long as the therapy is maintained, that is. I will shift gears here to more complex disorders that have really sort of been on our mind and mind of people, in the primary the field, the inborn error of immunity field for the longest time. This is the hyper-IgE syndrome. And my first actually encounter with patients with the hyper-IgE syndrome was back when, in the late 1980s, early 1990s at Boston Children's Hospital. And these patients have very high IgE levels. They have recurrent infections, and they have other non-immunological manifestations such as, bone osteoporosis. And there has always been a question mark about how the mechanism of disease is happening in these patients. Meneghish's group and Holland's group back in 2007 described loss of function mutations in STAT3 as an underlying genetic mechanism in the autosomal dominant form of the hyper-IgE syndrome. And since then, the thinking is, how would loss of function STAT3 mutation promote a hyper-IgE phenotype? Hyper-IgE syndrome comes in different flavors. So the autosomal dominant form, which is related to STAT3 loss of function mutation, is sort of accompanied by different forms, including TOC8 deficiency, which originally was described as autosomal recessive form of hyper-IgE syndrome. TOC8 deficiency is similar to STAT3-related hyper-IgE syndrome, the autosomal dominant form, in that both forms have very high IgE levels, have recurrent staph infection and bacterial infections. But they also differ in some significant ways. For example, TOC8 has problem with skin viral infections in particular, and viral infections in general, that patients with the autosomal dominant hyper-IgE syndrome don't have. So, and since then, other mutations in pathways like IL-6 receptor pathway and others have been associated with a hyper-IgE phenotype. So what brings all of these things together? And if you look at TOC8 itself, TOC8 is a G protein exchange factor. And essentially, it allows G proteins to move from being bound by GDP to GDP. And this activity is localized in so-called DOC homology region 2, or DHR2 region. And this sort of picture of how TOC8 works came very early after its discovery as a cause of the autosomal recessive hyper-IgE syndrome. And the idea is that it's really an actinopathy. And this TOC8 is regulating small G protein activity, making them active, such as CDC42. And CDC42 is a regulator of the actin cytoskeleton, and so TOC8 Similar to the WASP WIP system, DOCK8 is actually a regulator of the actin cytoskeleton, and its deficiency then is resulting in this disease phenotype. There's one problem with this idea, which is that some actinopathies are not associated with the phenotype of hyper-IgE or DOCK8 phenotype. And so there's always this nagging idea of what is it about DOCK8 specifically, aside from being a regulator of the actin cytoskeleton, that's giving rise to the phenotypes that I've just described to you. And so some years ago, Sevki Kellis in my lab worked on a patient with DOCK8 deficiency who had a unique mutation. And this mutation sort of inactivated the G exchange factor activity in DOCK8, but left protein expression intact. And so the protein is expressed, but it doesn't regulate the actin cytoskeleton and doesn't activate the small g proteins. This is the same here. And what she found is interesting is that this defect, and in fact, in general, DOCK8 deficient patients have a problem with activating STAT3. And in these patients, their cells, when stimulated with things like IL-6, the STAT3 doesn't translocate to the nucleus and doesn't activate gene expression that you would expect with STAT3 activation. And so this sort of brought the idea that both DOCK8 and STAT3 mutations have something in common, which is the regulation of STAT3 activation. And we dived a little bit further along that and looked at other mutations. And in fact, the mutations that we normally associate with the hyper-IgE phenotype, including IL-6 receptor mutations, DOCK8, STAT3, zinc finger 341, PGM3. These are all mutations that people have described to give a hyper-IgE phenotype, and all of them sort of cluster around this STAT3 pathway. So looking further into other patients with missense DOCK8 mutation, this part of the talk onward is not yet published, submitted. We found 2 patients with DOCK8 deficiency that had missense mutations. As a background, patients with DOCK8 deficiency usually have complete loss of protein expression, and rarely do we find missense mutation. Usually it's deletions of the gene that we see. These 2 patients had 2 different missense mutations that map what's called in the linker domain, in the area between the DOCK homology region 1 and DOCK homology region 2, the latter being the Jeff activation domain. The protein expression in these patients was normal. However, of course, they had the classical DOCK8 phenotype of hyper-IgE and severe infections. And when we looked at STAT3 activation, what we found is that, in fact, both of these patients has reduced phospho-STAT3 activation when their cells were stimulated with IL-6, again indicating there's a problem with STAT3 activation common to these patients. Now, if you look at their actin cytoskeleton activity, what we found is that, unlike other patients with DOCK8 deficiency, these patients had apparently normal actin cytoskeleton activation. So what you're looking here at staining of fibrillar actin with phyloidin after stimulating the cells with an anti-CD3 antibody. And in here, the orange and the pink lines are patients with complete DOCK8 deficiency. The 2 patients with the missense mutations were able to activate the actin cytoskeleton normally or near normal. So the phenotype can be segregated into a STAT3 activation versus an actin cytoskeleton assembly. And if we looked further, we found that actually DOCK8 not only is activated by when you have signaling via the T-cell receptor or IL-6, it translocates to the nucleus, and it translocates to the nucleus together with STAT3 as a complex. And these mutations that I described to you actually interrupt that translocation. And you see that both in cell lines, and you see it in primary human lymphocytes of patients with the respective mutations. So what's the nature of this interaction between DOCK8 and STAT3? What we could find is that you need both STAT3 and DOCK8 for either one to translocate to the nucleus. So if you have, for example, STAT3 deficiency, STAT3 will not obviously go to the nucleus, but also DOCK8 will be severely limited in translocation in the nucleus. These two seem to be bound in a complex. And looking more into this complex, we found that normally at steady state, without any activation, DOCK8 and STAT3 are associated and you can co-precipitate them. Different mutations do different things to this interaction. Some mutations, like this mutation, the H962, makes the interaction even much stronger. So you have more interacting complexes and more precipitation of DOCK8 and STAT3 when you precipitate with an antibody to one of them. Other mutations have different activities, maybe either less or no different from wild type. But in all cases, you have a defect in phosphorylation of STAT3. And if you look at where the interaction is taking place, it is primarily at what's called the interlinker domain between DHR1 and DHR2. This is where STAT3 seems to be interacting most with DOCK8. And if you look where on STAT3 the interaction is taking place, it's taking place primarily in the DNA binding domain and the N-terminal domain. And we looked further and we could demonstrate that there's actually a trimolecular complex. There's a JAK kinase, STAT3, and DOCK8 all come down. And the mutations affect these interactions differently. Some mutations don't interfere with the interaction, and some mutations, like this one, the 962, make the interaction stronger, even while all the time the DOCK8 mutations inhibit the phosphorylation of STAT3. So the scheme we derived was that DOCK8 and STAT3 are bound together at steady state. When you have a signal, they interact with JAK kinase and allow for STAT3 to be phosphorylated, and then the complex jumps to the nucleus. In DOCK8 deficiency, this trimolecular complex, either its formation, if you don't have DOCK8, or the phosphorylation of STAT3, if you do have a mutant DOCK8 expressed, this trimolecular complex formation does not result in STAT activation. And so, to go further into the next step, what is then the effect of this trimolecular complex formation and activation on things relevant to DOCK8 deficiency, like, for example, the hyper-IgE, the mucosal inflammation? And we made mice to study this thing at a greater detail. We put some of the mutations I described to you, including this mutation, which selectively targets STAT3 activation, and the second mutation, the 1987, that Sevgi Kellis originally described, that targets the activation of DOCK8 JEF activity. And if you look at these mice, all of them express the protein. The 962 was slightly less expressed, but otherwise quite well expressed. If you look at the actin cytoskeleton activation, you will see that the actin cytoskeleton is not affected by the 962 mutation, whereas the one mutation that affects the DHR2 domain, the JEF activity, that mutation completely abrogates the actin cytoskeleton activation. Again, indicating that you can segregate with these mutations the cytoskeletal function of DOCK8 from other functions. And if you look at the transmigration through a filter, again, the two mutations seem to segregate with the DHR2 domain mutation, the JEF inactivating mutation, resulting in incapacitation of the lymphocytes to go through a membrane. So if you look at STAT3 activation, both affected STAT3 activation. And if you look at translocation to the nucleus, both mutations affected translocation to the nucleus, both of STAT3 and also of DOCK8. The phenotypes could be different. So, for example, if you don't have actin cytoskeleton activation, like with the 9A7, you see the DOCK8 clustering in the cytosol, unable to go to the nucleus. But make a long story short, both mutations impacted the capacity to phosphorylate STAT3 and to translocate it as a complex with DOCK8 to the nucleus. So, sorry. The last part of my talk, and I'll make it quick, is that what is happening in terms of the hyper-IgE, what we think is happening, relates to what's going on in the gut, and particular in the role of STAT3 in the formation of immune regulatory cells of the ROR gamma T type that seem to be very important once formed to regulate allergic responses in the gut. These cells are formed very early in life, especially during the weaning time, and they are long-lasting. And there's been loads of recent work showing that these cells are actually critical to the regulation of tolerance to foods and to bacteria. Their deficiency results in allergic responses to both. And formation of these cells is part of what's called what we call ROR gamma regulatory circuit, involving both the T cells, the immune regulatory cells, the Tregs that express ROR gamma, and also antigen-presenting cells that also express ROR gamma. So they act as a unit. And we have shown before that if you actually delete ROR gamma in Treg cells, you get a very strong allergic response and IgE responses to food. So we did this genetic model where we deleted ROR gamma transcription factors specifically in regulatory cells, and you end up with very high IgE, very high antibacterial IgE, antibacterial antibodies, so both to food and to bacteria, suggesting again how important this pathway is for tolerance. And in DOCK8 deficiency, what you find is that there is decrease in the formation of ROR gamma Tregs and in the formation of antigen-presenting cells as well. The deficiency in ROR gamma Tregs is both in the mesenteric lymph nodes and in the lamina propria, in the small intestine and large intestine. As I told you, also the antigen-presenting cells involved in ROR gamma formation, they are decreased in these mice. And they are also decreased. This defect is also found in patients with hyper-IgE syndrome. So what I'm showing you here is staining from the peripheral blood of patients with hyper-IgE syndrome, both with STAT3 loss of function, DOCK8, PGM3. They all have profoundly decreased ROR gamma Tregs in their circulation, indicating that this is a common attribute of the hyper-IgE syndrome. So what is that imparting in terms of tolerance induction in these patients? So what we think is happening is that there is a loss of the formation of these cells in patients with hyper-IgE syndrome relating to their requirement for STAT3 to be activated for these cells to differentiate. And that this defect results then in mucosal inflammation and in the formation of IgE antibodies to gut content, both bacteria and food, suggesting that at least in part the hyper-IgE response is derived from this sort of mucosal dysregulation in these patients. And to expand on these conclusions, so this DOCK8 STAT3 activation favors mucosal tolerance, that there's a defect in this regulatory subunit in patients with hyper-IgE syndrome. And I didn't discuss today, but the DOCK8 STAT3 interaction I described to you is not really unique, neither to DOCK8 nor to STAT3, but different DOCKs and different STATs we are finding are engaged in such interaction, suggesting that what we're dealing with actually is a cytokine signaling network which can be then targeted by different mutations and diseases such as DOCK8, DOCK2, DOCK11, and the different stats. And these are the people who did the work, and this is work over several years. Much of the last part of the work was done by Xian Chen in the lab. And I also would thank my clinical collaborators who provided us with patients for these studies. Thank you.